Hello everyone, I am Dr. Sokolov and I'm very excited to be your instructor in advanced pharmacology for nurses this quarter. So this is the very first module and we're going to talk about basic concept and background in pharmacology. These are very important module in order to understand, the drugs you're going to use in the hospital. You need to go through to this basic concept first. So I hope you're going to pay attention to what I'm going to say and let's get started. So first of all, what is pharmacology? Pharmacology is the science of drug action on biological system. So when scientists are working on developing a drug, the first thing it is just a chemical that they have to try on different systems. So they can start, trying on first like cell lines, and then they will move on to small animals and test the drug to see what is the effect of that drug on those biological systems. But of course, as nurses, what you want to know is what are those drugs doing in a human body. So that's what is called pharmacology. This is the study of drug in humans. And so as you can see on this graph, so pharmacology is really the intersection of different science. It's an intersection between the chemistry and the biochemistry. So that's the knowledge of the source, the chemical properties, the biological effect, and then the physiology and the medicine, because you have to know what is the physiology of the system that the drug is going to work on, and then on a medicine point of view, you want to know what is the drug used for certain medical condition. So this is the therapeutic use of the drug. So there are a lot of confusion among students about, what exactly pharmacology is. So don't be confused. Pharmacology and pharmacy is not the same thing. Pharmacy is a separate profession, which is responsible for the preparation and dispensation of medication. So once you're going to work in the hospital, you're going to see that the pharmacy usually is located in the basement, and the pharmacists are the one who are going to prepare all the medication that are going to be sent to the floor, and the nurse is going to be the one who ultimately is going to give the medication to the patient. But the pharmacy and the pharmacists, they are in charge of preparing those medication and reviewing the prescription that is sent from the physician. So pharmacology, no, it's not where you study to be a farmer. So what are the difference between drug and therapeutic? So a drug is defined as any chemical that can affect living processes. So as I said, a drug can affect some cells on, in vitro cells, cell cultures, can also work on, small animals in, the pharmaceutical industry. Small rodent like mice and rats are often used, to test drugs first before administrating to human and getting the approval for human use. So that's what the definition of drug is. Therapeutic is the medical use of drug either to diagnose, prevent, or treat condition. So most of the time, of course, the drug is used to treat a medical condition. We will learn this quarter like different classes of drugs, and here I'm giving an example, which is a beta blocker, and beta blockers have different indication. One of those are hypertension, cardiac heart failure, and so this is the difference between the drug and therapeutics. So of course, in this course, therapeutic is our principal concern. But before getting to that point, we have to go over some basic concepts. What are the therapeutic objectives? When a drug is developed as a therapeutics, and I think, now that we are in the middle of a pandemic, you hear a lot about development of therapeutics and vaccines. So the therapeutic objective is to provide the maximum benefit with minimum harm. So you want to have a drug that is going to treat the condition. So here, if we talk about COVID-19, you want to prevent the replication of the virus in the human body and treat, the infection caused by COVID-19 with the minimum harm. Of course, you don't want, to kill your patient and kill the virus at the same time. So that's what the FDA is looking when there is an application for a new drug. They want to look at the benefit of those drugs and the adverse effect that can occur with those new therapeutics. So what are the properties that, again, the FDA is looking when they receive a new drug application? So they want to make sure that the drug is effective, and they want a drug that is safe. And also, you want to have a drug that is selective, which means if you're looking for a drug that works against coronavirus, you want to make sure that the drug is going to work specifically on, blocking the replication in the virus, and you don't want that drug to work on other, like on system of the human body and block the replication of some cell in the human body. So that would be what we can call the selectivity, if, a drug work essentially on the target that it is made for. Effectiveness, a drug elicits the response for which is given. So again, here is another example. ACE stands for ACE inhibitor. So this class of drug is used for the treatment of hypertension. So it's used to lower the blood pressure. BP stands for blood pressure. And an example of a drug among that class of drug is enalapril. So the response that you're expecting when you give enalapril to a patient, you want to look at the blood pressure. So you want to see if you have a reduction of the blood pressure once the patient is on that medication. So that's the most important property a drug can have. And in order to be approved by the FDA, a drug needs to be proven effective. And so that's why you have clinical trial, because there was a lot of attention in the media about hydroxychloroquine that may have been, a drug used for the treatment of COVID-19. But in order to be sure that the drug work on COVID-19, the ideal is to do a randomized clinical trial. And we're going to talk about those trials in the upcoming slides. So the other property that is very important is the safety. Of course, the ideal world would be to have a truly safe drug that has no harmful effect, even at high dose and at long duration. I don't know any drug that qualify as that safe drug. Any drugs can have some adverse effect, but some of those adverse effects can be very mild, and some other can occur with, when you give like a higher dose, you can see those adverse effects occurring. And you can also see some of those adverse effects with the duration of the treatment. The longer you use a drug, the more likely you are that those adverse reactions can occur. For example, acetaminophen, your Tylenol, it's pretty safe. If you use it, you have a headache, you take, 1 or 2 tablets a day for 1 day, it's really safe. No, for a patient who has chronic pain and use acetaminophen on the long run and keep increasing the dose, acetaminophen can cause liver failure. And so that's why there are guidelines where you don't want to give more than, let's say, 2 grams of acetaminophen per day. Because if you use 2 grams per day on the long run, you can see those liver failure occurring in those patients. Now, what is a selective drug? So what is selectivity? Selective drug elicit only the response for which it is given. So I give the example of COVID-19 antiviral. And so, of course, a selective drug would be a drug that has no side effects. So that means there is no unintended effect that is occurring within the therapeutic range. Again, this is the ideal world. There are no drug that have no side effects. Again, the severity depend on the drug and the dose. And so here are examples that are, those side effects that are not really, specific, like nausea, vomiting, headache, that you can see, um, with some drug, and that are not necessarily related to the mechanism of action of the drug, and that can occur even within the therapeutic range. Other properties that are important, in the development of the drugs are the reversible action. So most of the drug has a reversible action. So once they bind to their target, they bind, they act, and then the effect of that binding is reversible. And some example that is more that is easier to understand when you describe reversibility is to look at anesthetics. So if you give an anesthetic to a patient to put them asleep during a surgery, you know that you want that effect to be reversible. You don't want your patient to stay asleep for hours and hours. Other drugs, Viagra, I think everybody knows what Viagra is for. Same thing, the effect has to last and disappear once the half-life of the drug occurs. Now, antimicrobial. In that situation, you don't want reversibility because you want to kill the bug that is bothering a patient. So in that situation, antimicrobial, they would bind to the bacteria that is infecting the patient and will kill the bacteria, and the effect is irreversible. Once you kill the bacteria, they're not going to come back to life. They're going to have other bacteria that can keep growing, but the ones that are dead, they are dead. Predictability is like the patient responds to the drug. When you give a drug to the patient, you want to know what to look for. So if you're looking for, again, a drug that treats blood pressure, you give them medication, you measure the blood pressure, you see a reduction in blood pressure. You want, to have those predictable effects, and that's part also of, the safety of the drug. The ease of administration. This is very important to improve the compliance of the patient, because we all know taking multiple pills a day and or if you have like different drugs that are prescribed for a different condition, the ideal is to have one drug a day for each of these condition, and that will make the compliance better, because patients tend to forget, have I taken my medication this morning, and if they have to take it like 3 times a day, you increase the risk of having your patient not compliant. Freedom of drug interaction. Again, when you have a patient that has severe hypertension and that can be treated with just one medication, they're going to have a couple. Another example would be patient with HIV/AIDS. They can be on multi regimen, and you want to minimize the drug interaction between those prescribed drug for the condition of the patient. Low cost, very important these days, and that would be more, for the insurance and decide, how they are gonna determine the copay of those drugs. The more expensive they are, the more likely you're gonna have to pay more out of pocket. Chemical stability, also important, because if you have a chemical that degrades, that doesn't have a long shelf life, that can be problematic, because that would mean the pharmacy that has to store those drugs would have to make sure every time that they discard the drug that have expired. So chemical stability is important, but I would say it's more important for the people who are manufacturing, and how they're gonna provide those medication. Again, you are hearing in the news about the new COVID vaccine, and you heard that the vaccine that is approved, the vaccine that was developed by Pfizer, need to be stored at below freezing temperature. So it has to be stored at minus 80 Celsius. And that can be a problem because not all of the physician office, not all of the medical center have those type of freezer and don't know exactly how to handle those vaccine that has to be stored under such low temperature. And then possession of a simple generic name, it's easy for the patient, but it's also easy for the physician, the nurses, and the pharmacist. And especially for patient, they go to an emergency room and they are asked, oh, what are the drugs that you are using? If those names are very complicated, it's unlikely that they're going to remember uh, the name of their medication. Now, chapter 2, application of pharmacology in nursing practice. So, what is going to be your role as you become a nurse about those medication and how you can ensure that your patient is safe. There is, and about that, the evolution of the nursing responsibility regarding drugs. So it used to be 5 rights of drug administration and became 6 rights, which is giving the right drug to the right patient at the right dose via the right routes at the right time. And now you need to have like the right documentation. You have to know where to find the right information to provide to the patient in case they ask you a question about the medication, and you need to be educated about those medication, those drugs. In the application of pharmacology in patient care and patient education. Remember, you're going to be the one at bedside, and you may be the first one to see a patient when they come to the hospital before they even been seen by a physician. So you have to understand what treatment they were on. And I would say, what is maybe more important is at discharge. If they get new prescription, you are to be able to educate them about, their new medication and the importance of being compliant if you don't want to, if you don't want them to be back to the hospital, a couple of weeks after discharge. So what are your responsibilities? As I said, the responsibility of nurses has evolved over time. And now, as I said, you are involved in the pre-administration assessment. You have to know, what are their medical conditions? So are they getting the right medication? You have to be able to teach them the dosage and the administration. Of course, you're going to get your order and you're going to give, the medication as it is written on the order, but you have to teach your patient when you discharge them about how, to take that medication and how often they have to take it. And then when you are at the bedside, it's very important that you can evaluate the therapeutic effect and help them, again, when they're discharged to be adherent. So you're at bedside, you want also to look at adverse effect, toxicities, and make sure that with your team of physician and pharmacists, that this was really set, that they got the best dose based on their medical condition, age, ethnicities. And so that's why your role is very important in the hospital setting, but also, if later on you decide to work in a family practice, it's very important that you're going to have to be able to provide them those information. So by knowing the major adverse effect that drug can produce, you will know, what are the reactions that are likely to occur, and early sign that an adverse effect is developing, then you can know like what is the intervention that you have to do to minimize the discomfort and the harm. The physician is not going to be, at bedside all the time, and sometimes the decision and the intervention has to be done even before the physician rushed. And so you have to know, those signs of early adverse effect. And you have to know also which drug may interact with each other. We'll talk later about CYP interaction, so the cytochrome P450 interaction, the inducer and the inhibitors. I'm not going to go in detail right now, but we'll have a module about that. And then also look for diminished therapeutic effect or intensified adverse effect based on those drug interactions. So let's move on to chapter 3, which is drug regulation, development, and name. So with this pandemic, there is a lot of attention on drug regulation, development, and name, and I'm sure you're pretty interested in knowing how that works. It started, a while ago when the Food, Drug, and Cosmetic Act was the first legislation to address drug safety. It was like in 1938. And then in 1962 was another important step by the Harris and Kefauver Amendment to the Food, Drug, and Cosmetic Act that required a proof of effectiveness before new drug could be marketed. And this remains against now. And that's why, there are a lot of clinical trials that fail because when they analyze the data of those clinical trials, they cannot show that the drug is more effective than a placebo, and therefore the FDA is not going to approve the drug if the pharmaceutical company is not able to show that the drug is effective for the condition that they had the trial for. So what are the different stages of a new drug development? The first step is the preclinical testing in animals. So these are what is called non-clinical studies. They are not done in human subject. They are done in the lab. As I said earlier, they are first tested on cell culture, and then based on the response that they learn from the cell culture, they're going to move on in a living system in a model that is widely used in pharmaceutical research are the rodents, so the mice and the rats. And so they're going to test, they're going to look at safety in those animals and toxicity, because you don't want to give a drug to a patient that was never tested before in cell culture or animal model. Those clinical testing, can vary between 1 to 5 years. And we know now with the vaccine that this year was, you know, um, amazing how fast, research moved quickly to be able to develop new therapeutics and new vaccine. What you have to remember too is like the drugs or the vaccine that were developed, there was some prior testing that were done for the SARS-CoV-1, which is, another strain of coronavirus that affected the Middle East before. And then, based on the knowledge that they had from the development of those vaccines that never came to light, they had a better understanding on what to do to speed up the development of a vaccine for COVID-19. Similarly, they had a bunch of antiviral that were on the pipeline to treat Ebola, and those drugs then became available and be tested on COVID-19 model, and then move on to the clinical testing in human. So the next step after the preclinical testing is to move on to the clinical testing into human trial. And so you see this can vary between 2 to 10 years. So when you see that a drug move quickly within a year, this is why it's called, emergency use authorization by the FDA. They just give the authorization that would have taken more time to be approved, but because the drugs or the vaccine is so promising, there was like promising data with either the vaccine or the antiviral or the antibody cocktail that led the FDA said, okay, we're not going to wait any longer because of the emergency of the situation, but we are confident that there is enough evidence to see that the drug is safe, first of all, and then is effective. So after so first you have an investigational new drug application that is sent to the FDA. If that goes well and they have enough evidence, then the FDA will say, okay, now you can move on and you can start your clinical studies. So that starts after the investigation new drug application get approved by the FDA. And the first phase start with healthy volunteers. So it's a very small study between 25 and 50 patients, and in which they're going to look at the metabolism of the drug and the effect and how safe it is in human. So again, if you look at the antiviral for COVID-19. They have a very small sample of healthy volunteer, which usually are on the younger side. They're like in their 20s, 30s, a lot of medical student, or maybe like you may have no interest in, be in jumping in one of those clinical trials to help the science and help developing new antiviral for like COVID-19. So usually these are done in young healthy volunteer, and so that's why there is sometimes questioning when the target population is more at risk is older. How is it going to be when we switch from a healthy volunteer population to a population at risk who is older? And so once phase one, is promising, safety data looks good, the FDA give the approval to move on to the phase two. So phase two, that's the first time. So if we're talking about an antiviral, let's say it's an antiviral that is against COVID, you're going to have the drug, like let's say remdesivir, they're going to give the drug to a group of patients who receive the standard care, which, for now we still don't know what is the standard care. And then the other group are going to be the group treated by with remdesivir. And it's still like a small group of subjects, between 100 and 200. And they try, based on the safety data, they may still evaluate like two different dosage, like maybe a lower dosage for elderly patient and a stronger dosage in more like young folk, and see, if there's a difference in the dosage that is given in terms of, when you look at a disease like coronavirus, you want to look if at the stay in the hospital, and you want to look at the death rate. So if you compare the two groups, do we see an improvement in the length of stay in the hospital? Do we see a reduction in the number of deaths? So that would be an indication if the drug is working. Of course, you have the symptoms. You have a reduction of like cough. Respiratory distress, and so on. And that's how you can determine the rate. And then if this is looking good, again, move to phase three. Now, when you're in a pandemic, those two phases, phase two and phase three, were merged together, and they kind of had like interim data after some of the patients that were, randomized in the phase three, in the phase two cohort. They look at the interim data, but actually the phase three was ongoing with phase two just to save time. They didn't have time enough to say, okay, we're going to wait for the result of phase two to move on to phase three. So they start both at the same time, but then they can look at the phase two data earlier, and then if the phase two data are still, safe, then they can continue with their phase three trial without having to wait for the phase two data to start phase three. And then phase three, of course, is to enroll a larger population of subjects, usually is a number of thousands. Because again, when you look at the clinical trial where you have, subjects that are well identified before you start, that has no comorbidities, because it's still, an issue, like ethical issue, who do you enroll in those trials? And you want to have, the most diverse population in terms of sex, race, age. When you're talking about a disease like COVID-19, that affects any age, but ultimately has a higher risk of death in the older population. So how do you target that older population? It would be like in your phase 3 where you can start enrolling more patients that are older. And then again, the outcome measure is to look at, benefit in terms of symptom, but also length of hospitalization and mortality reduction. And here again, I'm talking about um, like a trial for an antiviral for COVID-19. For vaccine, it's a little bit different because um, you want to prevent from the disease and you don't want to inoculate the vaccine the disease to the patient who are receiving the vaccine. So what they do, they just have a control group, a placebo group, and then you have the vaccinated group, and they're just going to compare like who is getting infected. And so that's how right now when Pfizer released their preliminary result, they see that there was about like I think 94 subject who got COVID-19, and then when they look at it, there was only 10 from the group who had been vaccinated. The other one were in the placebo group. That's kind of where they said, okay, it's 90%, it's about 90% effective. And then not only they look at, who got infected, they also look at the immune response and the level of antibody in the patient who were inoculated with the vaccine compared to the placebo to determine um, the efficacy. But one thing that we still don't know is like, is this effective over time? So it's still too early to know how long this response it is for. Is it lasting for 3 months, 6 months, a year? Because of course for a vaccine you want at least the vaccine when it's against a virus like the flu or coronavirus, you want that vaccine at least to last for a year, and so you don't have to vaccinate the patient like every 6 months. It would be like way too costly, and then I don't think people will agree to be vaccinated that often. So we know with the flu, we have to. It's recommended to be vaccinated every year, and of course in patient at risk for like elderly, and if the flu season is known to be severe, they even recommend to vaccinate every 6 months. But it's too early to know what's going to happen with the COVID-19 vaccine. And then the phase 4, which is, you see, voluntary, is the post-marketing surveillance. So once drug has passed phase 1, 2, and 3, then the pharmaceutical company submit a new drug application, the NDA, to the FDA, and then the FDA will decide whether or not this drug will be on the market. So as we've seen for COVID-19, the FDA has approved drug for emergency use until they have more valid data to support that those drugs will have the full approval. And then so the phase 4, the post-marketing surveillance, is to see, okay, now that we went from 1,000, 10,000 subjects that were in the trial to 100,000 to million of subjects who are going to receive either a drug or a vaccine. What are the safety data that are we collecting? How often we are seeing adverse effects, adverse reaction, and we've seen drugs, that have been approved by the FDA, and then after the phase 4, surveillance, after a couple of years, then the FDA retracted the approval because it turned out that the drug was not safe enough. So the benefits and the so the harmful effect was actually stronger than the benefit, and so they decided that they have to withdraw. That was the case with some NSAID. Like Vioxx, that was withdrew from the market by the FDA because of the increased risk of GI bleed. So here is the stage of a new drug development. So this slide is in French because I studied in French and I couldn't find something that is better explained that this image. So you see in the bottom you have the preclinical development, from the synthesis of the chemical in the lab to the in vitro testing on cell, and then maybe on isolated organs, and then after on small animals. And when this, look good and safe, and once the FDA review the data and approve it, then it will move to phase 1. So remember, phase 1 is the first phase in which humans are being involved in the testing of new drugs. And this is done in healthy volunteer. Then you have phase 2, which is the first time you have the targeted population. So, if you're testing a new antiviral, you will have the subject who has the disease and they will receive the drug or the placebo. Now, again, when you give a placebo, when it's a condition, that where the patient can die, you don't just give them the placebo. You just compare the effect of the new drug with the standard care. And, they receive placebo, so they don't know if they receive, actually the new drug or just the standard care, but they're also taking, if it's an IV or if it's a tablet, they will receive the same uh, formulation of the drug as, the one that actually contain the active ingredient. And then in phase 3, that's when you have a larger sample of subjects who are receiving that new drug, and when all the data, from the phase 3 are positive, and the FDA will review those data. The pharmaceutical company will submit their NDA to the FDA, which will review all the data and give them the green light to um, commercialize their new drugs. And, it can be in the United States or worldwide. And now what you have to know is like each country has their own drug and food administration. So you have, the FDA is in the United States, but like let's say in the EU, they have their own also drug administration for the entire EU, and each country also have their own drug administration, and will determine whether or not they want to approve that medication in their country. Now, this is a list of the drugs that were approved as of November 12th by the FDA Center for Drug Evaluation in Research. So as of today, there are like 42 new drugs and biological product that were approved, and some were innovative new product that has never been used in clinical practice. So, for example, remdesivir to treat COVID is like a brand new drugs, even though it was already like studied for other viruses. That's the first time it was commercialized and approved for emergency use to treat COVID. Other drugs were approved, but they are the same or related to previously approved product. And so if you click here, you will have the list of those drugs that were approved this year. And so you can see what they are used for. Treat virus, treat lung cancer, tumor. A lot, you will see that a lot of those drugs are approved to treat some type of cancer. But you also have, what is exciting for Duchenne muscular dystrophy. There is a drug now that was approved for the treatment of Duchenne muscular dystrophy. New painkiller. So you see drug for HIV, for sedation. This is um, the list for Parkinson disease, and you can at home you can click on all those different drugs and see what they are, how do they work, and so on. And then on the bottom, this graph is the annual novel drug approval between 2008 and 2017. And you can see that the number vary each year, but in the recent year, more drug got approved, and mainly these drugs are in the cancer field. There is like a breakthrough in understanding, mechanism of action of drugs in treating tumors. And what I want to point out is like those the listing that I've shown you doesn't contain any vaccine, allergenic product, blood and blood product, or any plasma derivative, cellular and gene therapy. These are approved by the Center for Biologic and Evaluation and Research. So the list I showed you is only from the CDR, Center for Drug Evaluation and Research. And so I showed you, the different phase of the drug development. Now I want to go through like different type of trial that can be done. So the gold standard in drug development is to conduct a randomized control trial. Because the randomized control trial, as I said, you would have a group of subjects who receive the drug, a group of subjects who receive the placebo. And when you say double blind, it means the subject doesn't know in which group they are, and the investigator doesn't know which which group or which drug they are giving. They don't know if they are giving the placebo, and they don't know or if they are giving the drug. So it's just to reduce the bias. And we know why you give a placebo and just give them the drugs when compared to the patient who don't even receive a placebo. Because the placebo, some patient, you have just the placebo effect. They're going to take a drug, they're going to feel better. And then also those drugs or even vaccine, they have adjuvant that are either in the tablet or the vaccine, and you want to see if this contribute to the response or to any adverse reaction to the subject. So that's the most reliable way to objectively assess drug therapy, is using a control, a randomization. So and then blinding. So randomize, you don't choose who's going to get what, it's just randomized. And if you're interested to know what are the clinical trial available in the United States, but you can also look, I think, at trial all over the world. Right now, let's see if you're interested to contribute to the science and enroll in a COVID-19 clinical trial or vaccine. You can just type COVID-19, and here, you would look United States. And then if you're interested, California, oops, United States, California, city, Los Angeles, distance, let's say 100 miles, and then you can search. And you see 146 studies, but you also have, studies that are completed, and then you can see the ones that are recruiting, the ones that are, going to be recruiting. Here you can see that this one is at Ronald Reagan, recruiting COVID-19 recovered volunteer research participant for registry, progesterone for the treatment of COVID-19 in hospitalized men. They want convalescent plasma, so there are a lot of different trials. Now you can narrow down the ones that are for adults. Here you have both male, female. Study type, you want intervention, observational, let's see, you want intervention, clinical trial, study phase, let's say you want phase 3, and then you just apply. And in that case, you narrow it down to 44, and now you can see the one that are recruiting. Maybe we'll narrow down even more. And so here, phase 3, double-blind, placebo-controlled study of AZD, it's probably AstraZeneca, might be the vaccine. So they are recruiting right now, different type of therapy. And so if you want to know what they're about, you can just click on. And here you see this is 2-group phase 3 efficacy. You see it's a vector vaccine, so there's another vaccine that is in development, because you have to remember that so many doses are going to be needed to vaccinate the population, that just one single pharmaceutical company may not be able to achieve that goal. And so that's why the more vaccine available, the better it's going to be to control the pandemic. So let's go back to our slide. Now, what are the limitations of the clinical trial? So the limitation, of course, is who are enrolled in those trials. So now more and more women are enrolled in the trial, but there's still the question of, women becoming pregnant during a trial that, pose some concern in terms of ethical process of testing, women who become pregnant during the trial. And then, of course, same thing, children, a lot of ethical issues to test on children. Now, it always depends on what the condition is. Of course, you're going to have, studies, clinical trial in children when it comes to cancer treatment. But most of the time, phase 1 trial are going to be more likely targeting towards men, again, depending on the condition and the medication that is being developed. And so those patients also, if you go back, to the clinical trial website that I just showed you, if you look, the criteria of selection to enroll those patients, you're going to see that are very detailed. Even though they want people who mimic the general population, they also want to avoid some artifact that can be caused by the disease rather than by the drug. So they try to have, the more, I don't know if I can say pure population, but they want a population, even though that they can be as healthy as possible to try those drugs. Of course, if you talk about COVID, again, your patient are going to be sick with COVID, but they probably would rather treat someone who just have, COVID-19 rather than a patient who has COVID-19 and then has other comorbidity, even though they are the one more at risk of dying from COVID. In terms of knowing like the sole effect of the drug, they want to try on a population that would not interfere with the safety of the drug at first, and then they will move on to those patients who are more vulnerable to COVID, but ultimately they can be also more vulnerable to develop adverse effect with a new drug. So you have those patients that are carefully selected in the trial and may not represent the full spectrum of the population that is targeted. Also, again, those patients take the drug for a relatively short period of time. Most of the trial, I would say, would last like a couple of months. And again, it depends what you're looking at. If it's you want to look at the efficacy of antiviral, it's pretty quick. You want to kill the virus as soon, as fast as you can. If you are looking at a clinical trial, looking at vaccine efficacy, is going to take longer because we want to assess, the level of antibody that are produced by those vaccine over time, and how, this level sustain over time. But I would say on an average, they'll vary between 6 weeks and 12 weeks for most of the trial. And then again, another example, I work on Alzheimer's, and those trials are need to be longer because the disease process take long time, and also to treat a patient with Alzheimer's disease, a lot of the trial has failed because we looked at a 3 months type time points, and maybe you don't see an effect that quickly. But the longer the trial is, what you have to realize also is like the more money it costs to run those trials, but also it's hard to retain the patient in a trial that lasts longer because they have to be committed to come back and see their investigator to do like some check at each data point. If it's like after a month, after 3 months, after 6 months, after a year, you have to make sure that you can retain those patients in the trial because it's more likely you can have patient move, patient will die, and so the longer the trial is, the harder it is to retain the patient in the trial. Because the trial is done with those patients that are carefully selected, that or during a short period of time, you may fail to detect some of the adverse effect. And then again, I said the number is pretty small. Now, let's talk about drugs names. So there are like different type of drug names, and what you want to know, what you're probably worried, do we have to know all three names, the chemical name, the generic name, the trade name? I'm going to tell you no. You only have to know the generic name. If you can memorize the brand name, it's fine. But what we're going to see is like some generic name can be branded under like different brands in the United States and in the rest of the world. So I study pharmacy in Belgium, and the brand name in Belgium are different than the brand name in the United States. So if you're traveling and you are sick and you are asking for Tylenol in Belgium, they won't know what it is. And actually, I may have the example I'm citing, it's actually tricky because that may be one of the only generic name that is different between the United States and the international non-proprietary name. So in the United States, it's called acetaminophen, and so the brand name, as I said, is Tylenol. But the international non-proprietary name is called paracetamol. So if you go, even if you know the brand, the generic name of Tylenol, and you go to Belgium or France and you have a headache and you ask for acetaminophen, they may not know what it is. But that's an exception. Most like we talk about the ACE inhibitor and Alprel. This is the same generic name in the same international or the INN name, and the USAN names are the same. So if you know the generic name, and especially if you only plan of working in the United States, is what you need to know. You need to know the generic, and more and more the generic name, the generic drugs are used in the hospital, in the community, because they are cheaper. So only a few are still using brand name products, and so that's why it's important for you to focus on the generic name. So in terms of what are those different types, you have the chemical name. So here the example I'm talking about is Tylenol. So when it was developed in the lab, it was called the N-acetyl para-aminophenol. But you see, it's complicated. So when they move on to the clinical trial, then they said, oh, it's still better, even if those generic are still complicated. Let's just come with an easier name to remember, paracetamol, which is The international non-proprietary name is the generic name given to a pharmaceutical substance as designated by the WHO. And then the USAN, which is a unique non-proprietary name assigned to pharmaceuticals marketed in the US. So acetaminophen in that situation. But as I said, in 99% of the case, the INN and the USAN names are the same. Another, exception is salbutamol and albuterol, which are the Proventil, the ProHair. If you look at the brand name, so you see that for acetaminophen, they're like different trade name. In the US, Tylenol. In Europe, it's Panadol. In Europe, in Asia, and Africa. So that's why, if you memorize the trade name and you have international patient who are going to ask you about those medication through their brand name, you may not know what they are. And then albuterol, this is the active ingredient of your Proventil, the drug that is used for asthma. And in Europe, the trade name is Ventolin. So now which name to use, when, and why? So as I said, the problem with the generic, even though they are easier than the chemical name to memorize, they are longer. They have about, on average, 4.3 syllables compared to 2.3 for the trade name. So they're more difficult to remember. And then the problem with the trade name is like a single drug can have multiple trade names, as I explained earlier. Now, the same trade name can be used for different products, and that's, here I'm giving the example of Sudafed and Sudafed PE. Pseudoephedrine was removed from the market because, it's one of the ingredients of methamphetamine, and therefore, because the brand was so strong, when they switched to phenylephrine, they called it Sudafed PE. Now, what are the advantages between generic versus brand name? Generic products, it's like, are they equivalent? Are those generic and brand name product, are they therapeutically equivalent? Yes, in order for a generic to be approved, they have to show that they contain the same dose as the drug that is marketed under a brand name. And when the FDA approved those generic, they have to be considered equal. Now, there are, some drugs that it's hard to have the same equivalency, like therapeutic equivalency, because of the formulation. When you do a tablet, you have those active ingredient that contributes to how the drug is going to be released in the system. And so that's why you can have some slight variation. And so this is why there is an orange book which lists all the approved drugs with their therapeutic equivalent. So if you click on this link, you can search for drugs and see if they are therapeutically equivalent. For example, Synthroid, which one of the drugs that is used for the treatment of hypothyroidism. And this drug is known to have some issue with bioequivalency. So here they would list all the different type of active ingredient and their proprietary name, the dosage, and if they are bioequivalents. And so they are rated. There is this code that rate them. If they get an A, that means they are therapeutically equivalent. And if they get a B, that means they are not therapeutically equivalent. So when a physician is going to switch a patient to one of those because there is a shortage maybe of that specific drug, they have to look at the therapeutic equivalent, and they have to tell the patient, okay, you may have to adjust, you may see a difference in your response to the drug. And so you just have to monitor and stay in touch with the physician because they may need to adjust the dosing. And so, as I said, what is different is like the rate and the extent of absorption that can differ because of the active ingredient. And when we'll talk about neurotherapeutic index window, I will explain that on another module, but that's where it can be a concern because if you have a neurotherapeutic index, that means a slight difference in the response can be toxic and can be causing some serious adverse effects. So that's for those medication, it's really important to look at the therapeutic equivalence when a physician switch them from one to another. And we are at the end of this first module. I hope you appreciate it. And when I'll see you in class, we will ask any question you may have. Thank you.